And we were looking at epidermal cells, epidermal tissue of the plant body. So here you're looking at trichomes, and that those are just epidermal cells that have been elongated and or taken on different shapes. But basically, they come up off the leaf, and it can really help the plant deal with drier areas and helps them better adapt to drier areas where water isn't usually available all the time. It helps slow down water loss from their leaves and slow that whole process. And then, of course, certain trichomes can be glandular, for example, and they can have essential oils in them. And so, if you brushed your fingers across like the Cuban oregano that you saw in lab or some other herbs, you can definitely pick up the different scents of the essential oils. And those are primarily located in the trichomes. So here is another picture showing the waxy cuticle surface on the leaf. And then they have a nice image here of the two guard cells guarding the pore. And so the whole thing is called the stoma. Really the whole complex, the guard cells and the pore, they like to refer to as the stoma for singular, stomata plural. And so it does make sense that they do, coat their body with a waxy substance. On the above ground parts, it's the cutin wax. Below ground, even below ground, their roots will be coated with a waxy substance. That tends to be a different wax. And I think I'll mention that when we get into roots. I'll talk about that a little bit. But again, it's just to protect against water loss from coming. You don't want water vapor coming out of the cracks and crevices of the plant body. You want to really have good control on that. If you're a plant, you don't want to have that just happening because of cracks and crevices not being covered up. And so really they have tight control on water loss through the stomata. That's what they're using to allow for water loss. And if they close down those pores, then you won't have that water loss. The other thing that often doesn't get talked about, but it's still important to think about, is that these waxes can protect the plant against pathogens. Primarily, we're thinking more like bacteria and fungi. They have a harder time getting through that. Although certain pathogens definitely, they can be successful getting through it. No, yeah. So bless you. So the guard cells and the pore, really the way they labeled it, like they put stoma, I would have put the word pore there, and then I would have circled the whole thing and said that's a stoma, the guard cells and the pore. So yeah, the leaf, if you are a pathogen and you're trying to use the plant for food, the leaf tissue is pretty prime to want to try to chew and get access to the nutrients in there. So the plant has to deal with a lot of it. But one strategy can be that waxy covering. And then root hairs too. So root hairs are part of epidermal cells. They're similar to the trichome idea in the sense that they are epidermal cells. In this case, they're usually pretty typically just straight, elongated in a straight morphology, nothing fancy. But really super important role because it's the root hairs, and as you can see with this seedling, this is probably a radish seed, lots and lots of root hairs. And that's really, really important because it's the root hairs that are doing the majority of absorbing water from the soil system. And so the shape of the root hair, that long, skinny shape, really promotes a high surface area and really allows the water to be absorbed very efficiently. So if you've ever read directions, like when you look to, how do you repot a plant? And they'll say, take the plant and put it into a slightly bigger pot with fresh soil and then water well. And then if it's a good resource, they'll say, make sure the soil is consistently moist for several days. The reason they're telling you that is no matter how careful you are, when you repot a plant, you're stripping off a lot of the root hairs. And so now the plant has a tougher time efficiently absorbing water. And so they usually tell you baby it a little bit for a few days until it can regrow luckily it can regrow those root hairs. And so just give it a chance to get those root hairs established again before you, you can ignore it a little bit more. So really, really important. Okay, so epidermal tissue, similar in the same idea. It's the covering of the plant body, just like we have epidermal tissue on our body. And then one other thing I'll mention here, and then I'll talk a little bit more about it, especially when we get into stem tissue and stems in general. There's the paraderm idea. So as the plant gets older, so you think about a seed germinating and growing, and then we talk about it's a seedling, but eventually it starts to mature, get bigger. Then, depending on the plant species, you might get a paraderm replacing the epidermis. And so that will happen when you get further along in the age of the plant. And essentially, it's going to start laying down bark. And so the paraderm area is going to make sure that there's nice, healthy bark. Part of bark is cork cells, which are dead at maturity, but again, they're well coated with wax. And so these cork cells, even though they're dead and part of the bark system, they're really important for providing that barrier with the stem tissue so that you do get some nice hydrophobic surface. So suberin is the other wax that I would wanted you to think about. So on the leaves, you tend to get cutin wax. On the stem and root, you tend to get suberin wax. It's just a different chemical composition, but it's very hydrophobic. And again, it covers the surface of, in this case, the stem tissue. And then you can see the little white striations or dots on this tree trunk. And those are the lenticels that will allow for the stem to get easier access to gases, or essentially it's for gas exchange, I guess you could put it that way. And so you have these lenticels, these striations that you see on that tree trunk. It's just discrete areas where the cells are more loosely packed. So there's a fair amount of air gaps there. And so you have those discrete regions that do allow for more airflow in those regions. And that's important because, tree trunks that's all living, well, not all of it, but a lot of that tissue is alive and needs access to oxygen. And so you have to have a way for it to efficiently get that gas exchange going on. Okay. So yes, those lenticels are important for the stem to breathe, so to speak, get access to oxygen. And then here is what you would see under the microscope. If you took a tree trunk or a stem and cut it in cross section and looked at it under the microscope. So this area, you can see how those cells are more loosely configured. They're not so tightly packed. So that is a lenticel that you're seeing in that region. Okay. All right, the last couple of slides are some wonderful quotes from Michael Pollan's book and then also from Sir David Attenborough. So if you just want to read something beautiful, take a break from studying, the next two slides that I have in this slide set are a good thing to think about, read, and ponder. So please do so. But I'm going to switch over to the next slide set, which I think is posted in EDUCAT, called the plant body, right? Okay, good. So we're going to get into that now. And basically, what we're going to think about is the roots, stems, and leaves. And so the plant body, you have very different regions of the plant body that occupy different spaces in their environment. So we will start in the soil and work our way up. So we're going to start with roots. So here you're seeing a depiction of roots, stems, and leaves. The far left image is a root. If you cut a root down the length of the root and look at it under the microscope, that's a longitudinal section of a root. And so here, so the very root tip area, so you have to imagine that root is growing and extending by creating length because of the apical meristem. So the apical meristem is actually a little bit recessed, not right at the tip. So I think you can kind of see there's like this region or swath of cells that kind of look bigger, kind of elongated. And then you see a more smushed kind of section right here where the cells are more packed together, a little bit smaller. So once you start training your eye, you tend to pick up on that. But these bigger cells, that is the root cap, which is really, really important because soil is very abrasive. You don't want your apical meristem tissue to get damaged just by the soil, the frictional forces of the soil causing issues. So that root cap is super important. All right, so let's get into the roots a little bit more here. So when you take your seeds and you start germinating them, hopefully yesterday when you were in lab, you were looking at your seed from last week. Did most of them hopefully germinate? The mint should have worked pretty well, did it? Usually mint is hard to not have germinate. Usually germinates pretty well. But the first thing you should have seen out of any of those seeds, I don't care what species it is, you should have seen the radicle. And I want you to know that term. Okay, so the radicle is what we call the embryonic root. It's the root in the embryo. So when you crack open a seed, part of the seed is the embryo, the baby plant. Okay, and so the radicle is the first to come out. Now, a lot of people who don't know any botany, that surprises them. They're like, oh, it's not the leaves and the shoot, the above ground part, doesn't that come out first? It's like, no, when you think about it, most seeds, not all, but most seeds have some kind of food reserve in the seed. And so the really young seedling has a storage of nutrients to rely on. It doesn't immediately have to do photosynthesis to survive. However, it immediately needs water because seeds do not store up water. Seeds dry down quite a bit. That's a huge part of the maturation process of seeds is to dehydrate. Because if you have really moist seeds that have to make it through a winter before they germinate in the spring, fungi are going to be all over that. Fungi are going to smell that water, so to speak, and be all over that and destroy the seed. All right. So they don't have water stored up. So that's why the root comes out first, and the root's job is to go search for water immediately. So really nice imagery here of the timeline process of that. I think it's a radish seed germinating, growing. So really important, this part of the plant body. And again, the root hairs are really the major area for water and mineral uptake. And a lot of people don't know this either. The total root length of root systems of one plant can be several hundred miles. I know it sounds really weird, but it's true. And they've done the tedious math on this, the measurements and the math, and it's amazing. And so I think so many people don't appreciate this because what do we do when we experience plants? We don't look at root systems because root systems are underground. So we just don't really think about it that much unless you really get trained in on it. Most people aren't. So next time you're talking with someone who's not a botanist, give them some fun facts that really it's the roots that tend to be, that could often be a bigger biomass than what you see above ground. Right? So really, really extensive. Now, typically, this is what they'll tell you in terms of root development, that you have a taproot system possibility, or you might have a fibrous root system possibility. And they give you pictures like this that are very extreme examples. And that's true. However, I want you to realize this is kind of a continuum, all right? So you might have plants that have like a central taproot, but then lots of lateral roots coming off of it. And so there's a continuum between these two extreme examples. Root systems are way more complex than what most people realize. And so, but it's good to, it's okay to think about this in terms of, these are very good visual images to get you thinking about this central taproot with lateral roots coming off of it, with the dandelion being a great example. If you ever had to dig up dandelions in your parents' lawn, you know that their taproot is extreme. And then on the left, you can see a grass with its fibrous root system. So there's no central taproot. All the roots pretty much look the same diameter. And so that would be a fibrous root system. Okay, terminology I'd like you to know here. Okay, so you can see the artist drew the root cap a little bit easier to see visually with that brownish swath of cells here. So that's the root cap area, the root cells, and then kind of in that area where you see the pink cluster, that's kind of where you see a lot of the apical meristem cells. So very active cell division. These cells are dividing, and providing length to that root, okay, increasing the length. So at the very tip region, you have the root cap and the apical meristem. And then behind that, you see this so-called region of elongation. So these are some of the cells resulting from the apical meristem dividing, those cells dividing and dividing. So then those progeny cells, they start to elongate and get bigger. And then you see a little further back on the root is the so-called region of maturation. And if you were looking at roots under the microscope or the dissecting scope, look for root hairs, okay? So where you see root hairs, that's the maturation region, okay? Root hairs don't show up closer to the tip. All right, so the region of maturation. So I need you to know these regions, including the root cap, please. And then on the right, they're just showing like the different tissue types. So the so-called protoderm, you don't really need to know this, but the protoderm are cells that will eventually become the epidermis, okay? And then the so-called ground meristem, those are the cells that will become the ground tissue, like parenchyma cells, lots of parenchyma cells. And then the procambium, that's the region that becomes vascular tissue. So they give the baby embryo, they have special terms for what these cells are and what they're destined to become. All right. And so you don't, I'm not going to quiz you on protoderm, ground meristem, procambium. Okay, I'm not going to do that. But certainly talking about vascular tissue, parenchyma, epidermis, yes. But this is some botany terms that they use to talk about the baby, the embryo, before it matures out. Okay, here's a cross section of a root. And what they're trying to show is a very important process. How does water move once the root hairs take up the water? Then the goal of the plant is, I want this water to get into my xylem so that it can start moving through the plant body. Right? So it's got to get to the vascular tissue, to the xylem. Well, you can see the black line. It's trying to show movement of water, starting with, uptake by the root hair. And so you can see here, they have the water. What they're trying to show is, at least through this initial pathway, they're trying to show that the water is just skirting around all the cells. And so it's just flowing through the cell wall spaces. Right? They're showing it here too. It's just skirting around. So there's different ways that you can get water moving through the root tissue, and they're zooming in on one particular method, which is what they think is the most efficient. And so this method of movement is called apoplast movement of water. So that apoplast is just referring to the idea that water is moving in the cell wall. The water is flowing through the cell area. It's just skirting around the cells, not going into them at this stage. Okay? So apoplast movement. Now, so let's see. So it's happening in the epidermis region. All those yellow cells that you see here, that's all parenchyma cells. All right, that's ground tissue, or they often refer to it as the cortex, but it's just parenchyma. All right, so it's moving through the cell wall area, and then there's this band of cells right here. That's called the endodermis. That I need you to know. All right, so the endodermis is a ring of cells that is surrounding primarily the vascular tissue inside of that. Okay, and you see that they have they've blown out that ring of cells over here in figure B, and you can see this orangey band. So what they're trying to show is that the band So that's all in the cell wall, that orange band, that's part of the cell wall. And it's called a Casparian strip. Casparian strip. I was looking for the word. Casparian strip right here. So that red band is called the Casparian strip. It's a specialized hydrophobic strip, part of the cell wall. And effectively what it does is it forces water to stop moving through the cell wall area and dive through the cell membrane. So water cannot get past that Casparian strip. The water would, like, moving in the cell wall space, it would bump into that Casparian strip, and that Casparian strip is too hydrophobic, and the water would just be blocked. And so that forces the water to then get into the cell membrane, pass through the cell membrane, and get into the cell. So that's what they're trying to show here, is that you finally get water moving from cell to cell after it leaves the yellow area here where all the plankton. So I won't. There's a lot of like physics and physiology to why it does this. In a nutshell, why it created this situation, because initially it sounds like kind of a pain. Like it seems like just keep it going to the cell wall. What's the big deal? But essentially what it does is it forces water to stay moving in one direction only, which is keep going toward the xylem. Don't try to come back out. And again, there's a lot of physics and osmosis and stuff going along with that. But basically it's a way to keep water just going one direction from the soil to the xylem and not back out again. Okay. Questions on water movement? Okay, so let's see. We have some other pictures here too. All right. So another region, let me go back to that previous slide because they had it on there too. The pericycle. Let me show you on here. So the pericycle. The pericycle is another ring of cells just inside the endodermis. Okay. That is a meristematic ring of cells. So from the pericycle, once this seedling gets a little bit bigger, the pericycle might get activated and start to go into cell division. And it gives rise to lateral roots. So it allows for lateral root formation off of the root, okay, the initial root. And so that's another meristematic tissue that can be very useful for lateral root formation. So you're just seeing some different images here, trying to show the root cross section. And they have the endodermis, so this is an actual root cell, a little messier to see, but the endodermis, grain of cells, and ring is first. And so that parasite, I missed from the parasite. And you can see there's, a fair bit of tissue destruction along the way, but the end result is good because you end up with those lateral. So the pericycle is another meristematic. Yes. Yeah, I mean, essentially they're parenchyma cells. They're living, functioning, no secondary cell wall. But they're meristematic, whereas they're just always ready to divide if given the right signal. So yeah. Okay. Now we're going to be in this class, we'll be zooming in a lot to the angiosperms. And so I'm going to talk to you a little bit about the angiosperms of the flowering plants and what their root structures look like. Okay. So when you talk about flowering plants, the angiosperms, you divide them into two main classes. You say they're either a dicot or they're a monocot. Those are your two choices. By the way, if someone were to, if you're out hiking with someone and like, is this a dicot or monocot? And you have no idea, guess dicot. So there's more dicots out there than any monocot. Okay, so might as well just say dicot if you don't know. But I'll teach you how to clue in. So hopefully you'll have an informed opinion by the end of the class, right? So here you see the drawing up top of the two options of a dicot on the left and a monocot on the right. So what are they looking for? They're looking at the vascular structure, and that can actually tell you a lot. And so here they've taken a stem and cut it into cross sections, and they're looking at all the different parts of the tissue. And they zoomed in. If you can see the vascular tissue, they drew it here in blue and green up top. And you can see how the left, the dicot example, I say it has like a plus configuration, the blue area, plus or cross, that's the xylem. Okay, so that's part of the vascular tissue, the xylem. And so I say it has like a plus configuration. So you can see that here, the actual root down low. Let's see if I can pull this up a little bit. They have that cross shape. So the xylem, it looks red. All the cell walls are stained red because they stain for lignin, and lignin is in the cell wall of xylem because they have that really thick secondary cell wall. And then all that kind of blue-green that you see stuck in the corners of the plus sign or the cross, all those little blue cells, that's the phloem. So you have a cluster of xylem. It's not going to let me. You have a cluster of xylem. Yeah, it won't let me do it. Never mind. You have a cluster of xylem cells, and then you have a cluster of phloem cells. They're all, clustered together in one spot here. And in the dicot, you tend to have that very nice plus shape of the xylem tissue. Okay. Now look over on the right, up top, you can see we say this is a ring of tissue of xylem and phloem. All right, so the blue is the xylem, and it's a ring of xylem cells. And then right near that is a ring of phloem cells. And then the actual image down on the bottom right, all the bigger, those really big cells, that's part of the xylem. Those are vessels. And then the smaller red cells are tracheids. So remember, xylem has vessels and tracheids. When you look at angiosperms, vessels and tracheids. So you can see visually the really big ones, those are the big vessels. And the tracheids are smaller. And then kind of to my eye, it kind of looks bluish green. That's the phloem. Okay. And then the ring of on the right one, it's really obvious. You see the ring of red cells. That's the endodermis with the Casparian strip. Okay. So another thing to take note when you're looking for vascular tissue in a root or stem, you look, where is the vascular tissue with respect to the rest of the tissue? And so if someone were to just give me this picture and not have it labeled at all, and they were to ask me, what is this? Is this stem, root, or leaf tissue? I'd look at it and I'd look for where the vascular tissue is. And in both cases, whether it's the dicot or the monocot, do you see how the vascular tissue is pretty much in the center of this tissue? And then you have lots of tissue surrounding it, right? So I know from my training that you're looking at root tissue in this case, all right? Because when you have the typical root, they're buried in soil, right? So they're supported by all that soil packed against the roots. So it's okay to hide your vascular tissue deep inside. But remember, xylem in particular is pretty sturdy. And so if I have stem tissue, I will put my if you think a little bit like building a skyscraper, you don't put steel beams of a skyscraper smack in the middle and call it good. You have that scaffolding of steel beams around the perimeter of the building. And so stem tissue tends to have xylem further toward the perimeter. Root tissue tends to have xylem closer to the center. Okay, so that's kind of the physics of what roots and stems have to deal with in their day-to-day life. Okay, so I do want you to know root structure, please. So the anatomy this slide's a really good one to study and learn the anatomy of the different parts of what the roots look like in cross section like this, including being able to label like the xylem and phloem. Are there questions on basic root structure? Okay. A little bit about some specialized roots. So we said that when a root comes out of the seed, we call that the radicle. That's the embryonic root. That's the root that was destined to be a root right from the get-go. It was part of the embryo, the baby plant inside. That is in comparison to what we call adventitious roots. This is different now. These are not embryonic roots. Adventitious roots, they use that term when you get roots forming from pretty much any other part of the plant body other than the embryo inside a seed. Okay. So adventitious roots, if you've ever taken a cutting from a friend's plant, like, oh, I really want to, grow that plant, and you take a stem cutting and bring it home and put it into a cup of water, then you watch those roots form. Those are adventitious roots. And what you've done is actually started a process that is absolutely amazing that the plant has to accomplish. Because when you take a stem cutting and you cut a stem off of a plant, now that cut region of the stem has to go, whoa, I just got, amputated. All my roots are amputated. I have to regrow roots. And so now that stem tissue stops being stem tissue, morphs into root tissue, forms adventitious roots. I mean, that's phenomenal. We can't do that. You just don't have the capacity to do that. So next time you propagate a plant with a stem cutting, you should appreciate what a massive undertaking you just initiated by the plant. Now what the plant has to do. And they do it so well, and they do it pretty quickly too. So I'll show you a picture of some corn plants. If you've ever plucked corn, harvested corn by hand in cornfields, if you've looked down, there's prop roots on the stem that you can easily see above ground that are, again, the physics of it help keep that stem upright and not get knocked down by wind or you going through the corn rows. So basically, adventitious roots, those get formed a lot anytime you propagate plants. You tend to see they're going to start forming these roots. So we give them special names. We call them adventitious roots. So here's corn at the bottom at soil level, and you see those really funky looking prop roots, but very, very helpful. Okay. Here, these roots, these are specialized in the sense that they're aerial. They're aerial roots. Now, most plants that we're going to talk about in this class, we tend to think about roots buried in soil, and that's how you grow the plant. But just be aware, especially with orchids, most orchids actually grow on other plants, like on trees. Most orchids are tropical, and so most of those orchids, they grow on branches of tropical trees. They're not hurting those trees. They're just using them for support to get closer to the sun. And so they have their roots just dangling off their body. And so they're green. They're usually the epidermis is many layers thick, just to kind of beef it up to prevent water loss. And so velamen is often the term that you'll see used. That's a term that they use to describe these aerial roots that are green, and they tend to have multiple epidermal layers. Okay, so the roots can technically, if they have chlorophyll in them, they can do photosynthesis as well as the leaves. So orchids are really, really interesting. The orchids we have around here, by the way, they're not, I don't think there's any species up here that can do aerial growth. I think they have their roots in the soil. Lady slippers are the most common around here, pink, and I usually see pink lady slippers, but there's others. My one Carrie, a lot of you might know Carrie in the Indoor Ag and CEA program, and she did her whole work on ram's head lady slipper. in the Munising area, Pictured Rocks area. There's millions of them there, at least a couple million, which was stunning. We didn't realize there were that many there. Another picture. So of all the orchids out there, the only one that we use commercially is the vanilla orchid, where we get vanilla from. That's I guess that would technically be called a spice. But and then if you live further down south and having encountered banyan tree roots, those are amazing. So they actually grow down from the stems, the tree, those tree branches. And so the adventitious roots will grow down until they hit the soil, and then they'll start growing into the soil. And so they can be this massive growth of adventitious roots. Kind of funky. Okay. So roots, incredibly important. Next up are the stems. So stems are above ground most of the time, but not all the time. So there's some specialized stems that are actually, but let's think about the typical stem that's above ground. And so you will have an epidermis layer. Remember, that's going to usually have some suberin wax protecting it. You'll have a cortex region, which is just a bunch of parenchyma cells. All right, that's just a bunch of parenchyma cells that will form the majority of the stem tissue. And then inside of that will be the vascular tissue. So the cortex region, yeah, it's mostly parenchyma. Sometimes though you'll find parenchyma cells, yes, for sure. Sclerenchyma, yep, secretory cells, yeah. So, but the definite winner there is parenchyma. You have lots of parenchyma. Okay, so remember, parenchyma are living at maturity, but they are thickened up a little bit, and they tend to be toward the surface of stem tissue, like the celery stalk example that I gave you last week. Sclerenchyma, remember the pear tissue? When you eat pear tissue, there's sclerides in there, and then other sclerenchyma are fibers. So, but for sure, when you crack open a stem, the bulk of what's inside are parenchyma cells. All right, they're very important. And then if you go past all that parenchyma cells and get more toward the center a little bit, you'll see vascular systems. Well, I shouldn't say more toward the center. Not really. I'm going to show you a pattern. I'll show you what it looks like under cross section. All right, so you're going to see some of the same ideas that you see pericycle. You're going to see line phloem. I'm going to talk to you about the vascular cambium in a little bit more detail. We've introduced these terms last week, right? But now I'm going to show you what does it actually look like in stem tissue in more detail, right? And then the pith is just the word that they give for the very center of the stem. Some stems have a so-called pith region, and I'll show you that too. So all these different cell types, it depends on what plant you're looking at, the design. What's the overall design? What does it look like? Okay. And so it depends. They tend to separate like the gymnosperms, which, the conifer trees around here are all gymnosperms. And then all the so-called woody dicot angiosperms. So when we look at angiosperms, another way you can think about them besides monocot dicot is, are they woody at maturity? Do they have like bark and definite, the stem tissue is really beefed up with bark? Or are they no bark and no thick stems? And so woody versus like the other comparison would be like herbaceous, right? So these terms you'll start seeing a little bit more. And so the first category are the gymnosperms and the woody dicots. Another category are the herbaceous dicots. They have a different configuration, so all three of these, the third one, are the monocots. So they all, depending on if you're looking at plants that fit into the category 1, 2, or 3, how they arrange all that vascular system, it differs. And that, so you can use that as a tool. Like, you know, someone shows you a picture of something and says, what is this? If you start to understand how plants organize all of this, you can tell just by a picture in cross section what, you know, what it is you're looking at. So let's look at an example. So here's a gymnosperm stem. So this is like, you know, a red pine tree, white pine tree, something like that. In this case, it's actually a Pinus genus. So, but anyways, here you're looking at the pith. So in the very center is the pith. Those are just parenchyma cells. Mostly that's what they are. Okay? And then all the kind of reddish that you're seeing here, there's two big swaths of it. So these are growth rings of, and this is all xylem, most of it anyway. This is all xylem, all the reddish. Okay. So that's xylem tissue. And then the demarcation between the reddish xylem and then the kind of blue-green, that ring of cells, that is the vascular cambium. Right? So they have it labeled off to the right here. So we talked about this last week, how you have that ring of cells called the vascular cambium that's meristematic. And so it's going to divide. And when it creates, you know, when each cell goes through cell division, you end up with 2 cells, but one will be destined to become a xylem cell, and the other one will be destined to become a phloem. And so here, the kind of the bluish green right on the other side of the vascular cambium, that's all mostly phloem and is active. The older phloem further out is dying off. Remember, phloem is living cells. It only has a primary cell wall. And so the pressure exerted against some of the older phloem will die off. So you tend to see the buildup of xylem. Remember, xylem's super tough. Got that secondary cell wall. So that will persist. But the more fragile phloem will get crushed. And so you have a thin band of active phloem. All right. And that's why you never see this tons of phloem when you cut a tree trunk in cross section. You see lots of xylem, but not much phloem. Okay. By the way, you may have done this when you were a kid, but hopefully you won't do this after you learn this. Birch trees, you know how they have the bark kind of flapping off, and it's so tempting to take it and just keep peeling around. Please don't. If you do that too deeply, if that peel comes off in a circle all the way around, what you've effectively done is taken all the phloem off. And if you take all the phloem off in a concentric circle like that, you've killed the tree. And it's a slow death. So please don't do that. And my boys, when I take my boys hiking when they were little, oh, it's irresistible. You know, you see that flap of bark hanging and they would want to pull. And I'm like, no. So what you've done there is if you take that bark all the way off, all the way around, you have no phloem left. And so the sugars that get made in the leaves, they'll come coursing down the tree trunk to that spot, and then they can't go any further. So the roots never get fed. So then the roots start to starve. And if they starve and die, the whole plant dies. So it's an awful death. So please don't do that. It's called girdling. Have you heard that? If you girdle a tree, that's what you've done. Killed it. Very slow down. So don't do that. Okay. Vascular cambium, xylem to the inside, phloem to the outside. You see some resin ducts. You know, I think you're pretty familiar that some bark, especially you could poke at it and that those resins and saps come oozing out. Okay. So now they have another swath here that's labeled bark. Okay, so the definition of bark, if you look that up, they'll say it's all tissue outside of the vascular cambium. That's the definition. So that whole, everything from the outside the vascular cambium. So that means the phloem, because the phloem is right outside the vascular cambium. The phloem and the resin ducts and the cork cells and everything else that makes up bark in that kind of bluish green swath. That whole section is called bark. Okay. So gymnosperms Another thing to take note here, I think if you look, we're just focus in on all the xylem here, and you can see all those red cells, and they look pretty much the same diameter, right? Now look at this next picture. So look at the diameter of the xylem. Now look at here, where the red cells are here. Do you see all the red cells, the difference in diameter? Right? Some small, some big. That's a dead giveaway that you're looking at an angiosperm, because angiosperms have those larger vessels and smaller tracheids. So when you look here, all these bigger cells, that's all vessel, and then there's a layer of trachea. So what's going on here is in the spring, if you are a dicot angiosperm and you have the ability to decide, do I want to put vessels down? Do I want to grow vessels, or do I want to grow tracheids? In the spring, if it's been a nice winter and you have lots of water or snow melt, lots of rain, your choice would be vessels. If you're smart, if you're a smart plant, you grow a bunch of vessels. That's what you do, because that's very efficient at taking up all that lovely water from spring melt. Now, as the summer goes on and more drought happens, then you're going to go, ooh, I think I'm going to lay down tracheids. And so that's why you see different banding patterns. You see like soft vessels, then it transitions. It's because of water quality. And in a nutshell, if you have lots of water, go with vessels. It's very efficient. If you don't have much water to draw into your body, tracheids are a better bet. You have less issues with that in terms of getting the water safely moved through your body. I'll explain a little bit more about that as we get into movement of water through the body of the plant. So look for different sizes of the xylem, the vessels and tracheids. That will tell you what you got going on in terms of, is it a gymnosperm or is it an angiosperm? Okay. But again, you see there's this central pith region. Then you have, in this case, 3 growth rings. So 3 years of growth, where you see 3 years of xylem being laid down. Then you have the vascular cambium, a little bit of phloem, bark tissue. Okay. All right, we'll continue on with stem tissue and then get into leaves on Friday. We might be able to start algae and fungi on Friday. We'll see how that goes.